Alright, let's talk about MHC class 1 versus MHC class 2. What they are, where they are, what they present, who they activate, and how peptides get loaded. So MHC molecules are encoded by HLA genes, and their job is to present antigen fragments to T cells and bind T cell receptor. T cells do not see whole proteins, they see only short peptide fragments sitting in the groove of MHC. For MHC class 1, the loci of MHC class 1, which is HLA genes, the loci are HLA A, B, and C. So MHC 1 loci have one letter at the end, A, B, C, HLA A, HLA B, and HLA C. Its binding partner is CD8, TCR plus CD8. So CD8 into MHC1, 8 into 1, it will give 8. So it's just a quick matching trick. If you see CD8, think MHC class 1. About the structure, MHC class 1 has one long chain plus one short chain. The long chain is the MHC1 alpha chain. It has multiple domains, alpha 1, alpha 2, alpha 3, and the short chain is beta 2 microglobulin. Make sure you know that the peptide binding groove is formed by the alpha chain, while beta 2 microglobulin stabilizes the complex so it can sit properly on the surface. The expression of MHC1 is on all nucleated cells plus APCs, antigen-presenting cells, and platelets, except RBCs, because RBCs don't have nucleus. No MHC1 presentation. The function of MHC class 1 is to present endogenous antigens, example viral or cytosolic proteins, to CD8 positive cytotoxic T cells. Endogenous just means that the peptide comes from the inside of the cell. A cell is infected by a virus or a tumor cell with abnormal proteins that leads to MHC class 1 presentation and CD8 killing. And how does it load antigen? Where peptides get loaded? So the peptides are loaded onto MHC1 in the rough endoplasmic reticulum. It is delivered via TAP system. What is TAP? TAP is transporter associated with antigen processing. So inside the cell, proteins get broken down into peptides, often via the proteasome. These peptides need to get into the rough endoplasmic reticulum where MHC1 is being assembled. TAP is the transporter that moves these peptides into the endoplasmic reticulum. Then the MHC class 1 peptide complex goes through the Golgi and gets expressed on the surface. So if the question talks about TAP or rough endoplasmic reticulum loading, it is pointing to MHC1. For the MHC class 2, its loci, which is HLA genes, is HLA DP, DQ, and DR. MHC2, 2 letters, DP, DQ, and DR. The binding partner is CD4, because MHC 2 into 4, you get 8. TCR plus CD4, MHC 2 into 4, it will be 8. For MHC 1, it's CD8, 1 into 8. Okay, so if you see CD4 helper T cells, think MHC class 2. The structure of MHC class 2, it has 2 equal length chains, 2 alpha and 2 beta. MHC2 is made of 2 similar sized proteins, an alpha chain and a beta chain. Together, they form the peptide binding groove. The expression of MHC2 is on the antigen presenting cells only. Okay, so the antigen presenting cells usually means the professional antigen presenting cells, which are dendritic cells, macrophages, and B cells. So if the stem says macrophage injects bacteria, or dendritic cell presents antigen to naive T cells, that is classic MHC2 territory. What about its function? MHC2 presents exogenous antigens. For MHC class 1, it's endogenous. For MHC class 2, it's exogenous antigens. Exogenous antigens such as bacterial proteins to CD4 positive helper T cells. So, an exogenous means that antigen come from outside of the cell. So it got taken in by endocytosis or phagocytosis and then processed. How about antigen loading in MHC class 2? Antigen is loaded after release of invariant chain in an acidified endosome. Elaborate a bit more, MHC2 is made in endoplasmic reticulum, but it must not grab random endoplasmic reticulum peptides. So it's protected by the invariant chain while it travels. It gets trafficked into endosomes that become acidic, where the invariant chain is removed, and then the exogenous peptides can load onto MHC class 2. Then MHC2 peptide goes to the surface. The invariant chain leaves behind a small fragment called CLIP, clip, and a molecule called HLA-DM helps remove clips so the real antigen peptide can bind. So this is the knowledge that you need to know in USMLE preparation. So if you see acidified endosome or invariant chain, that is MHC class 2. So to summarize, MHC class 1 is HLA-ABC and CD8, and endogenous antigens, rough endoplasmic reticulum plus TAP, and beta-2 microglobulin, all nucleated cells except RBCs, the expression. Okay, MHC class 2, HLE is HLA-DP, DQ, DR, binds with CD4, and for the exogenous antigens, the antigen loading process is release of invariant chain in an acidified endosome, and it is expressed only on the professional antigen presenting cells such as dendritic cells, macrophages, and B cells. So how can this stuff show up in the question stems? So if you see something like a virus infects a hepatocyte, just pause and think, okay, viral proteins are being made inside the hepatocyte. That's endogenous, so that means MHC1, and the T cell that binds is CD8 T cells. If you see something like a macrophage, phagocytosis bacteria, that's literally the classic exogenous setup. So the bacteria came from outside, it got eaten, processed in vesicles, so that goes to MHC class 2, and the T cell that matches is CD4. The test can also check about pathway defects. For example, if the question mentions TAP, transporter associated with antigen processing, TAP defect, if there is TAP defect, what that really means is peptides can't get delivered properly for loading onto the MHC1 in the rough endoplasmic reticulum. So you can't load MHC1 well. So you end up with a weaker CD8 response. So TAP defect, think MHC1 problem, CD8 problem. Okay, if they mention something like invariant chain problem, that points to MHC class 2 pathway, because the invariant chain is involved with MHC class 2 trafficking and proper loading in the acidified endosome. So if invariant chain is messed up, you can load MHC class 2 properly and you get a weaker CD4 response. So invariant chain defect, think MHC class 2 problem, CD4 problem. And then there's this extra concept, which is cross presentation. So normally exogenous antigens go to MHC class 2 and activate CD4, right? But dendritic cells have this ability where they can take an antigen that came from outside, like something they phagocytosed, and still present it on MHC1 instead. The reason that matters is because it lets them activate CD8 cytotoxic T cells even when the dendritic cell itself isn't infected. That shows up in vaccine-style logic and some antiviral responses. So if you ever read a stem that feels exogenous, like dendritic cell takes up antigen, and then the question is clearly pointing to CD8, the way that can make sense is cross presentation, meaning exogenous antigen ending up on MHC1 to activate CD8. So the whole thing in one simple flow: inside the cell, MHC1, CD8. Outside and ingested, MHC class 2, CD4. TAP defect, MHC1 loading issue, weaker CD8. Invariant chain problem, MHC2 loading issue, and weaker CD4. And cross presentation is the special case where a dendritic cell can take exogenous antigen and it still present it on MHC class 1 to get a CD8 response. Next, let's talk about HLA subtypes associated with diseases. This is very high yield and they come in questions very often. For HLA B27, this one is linked to a group of arthritis, and the mnemonic in FA is PAIR. P for psoriatic arthritis, A for ankylosing spondylitis, I for IBD associated arthritis, and R for reactive arthritis. So if you see B27, think PAIR. Again, P for psoriatic arthritis, A for ankylosing spondylitis, I for IBD associated arthritis, and R for reactive arthritis. The next one is HLA-B57. This is upper cover hypersensitivity. If you see upper cover reaction, think about HLA-B57. Another one with a fun mnemonic is HLA-DQ2-DQ8. This is associated with celiac disease. The mnemonic is I ate too much gluten at Dairy Queen. 8 is HLA-DQ8, too much, 2 is HLA-DQ2, gluten, celiac disease, Dairy Queen, HLA-DQ. Okay, so Dairy Queen, DQ8, 8, HLA-DQ8, 2 is HLA-DQ2, and gluten is, you will think about celiac disease. Okay, so HLA-DQ2-DQ8 is associated with celiac disease. I ate too much gluten at Dairy Queen. HLA-DR3. HLA-DR3 is associated with type 1 diabetes, SLE, Graves' disease, Hashimoto thyroiditis, Addison disease. So the mnemonic includes two parts: diabetes type 1, diabetes type 1, diabetes mellitus type 1 is associated with HLA-3 and 4. So the mnemonic is 1 plus 3 equal to 4. 1 represents diabetes mellitus type 1. 3 represents HLA-DR3. 1 plus 3 equal to 4. 4 represents HLA-DR4. So diabetes mellitus type 1 is associated with HLA type 1 and 4. And another mnemonic is SL3. SL3. Regularly we write SLE with E, but you just reverse E to become 3. SL3, SL3. So it's meaning SLE associated with HLA-DR3. Okay, what are associated with HLA-DR3? Type 1 diabetes, SLE, and then Graves' disease, Hashimoto's thyroiditis, 2 diseases associated with thyroid, Graves, and Hashimoto's thyroiditis, and Addison disease. Type 1 diabetes, SLE, Graves' disease, Hashimoto's thyroiditis, and Addison disease associated with HLA-DR3. How about HLA-DR4? Okay, we have already memorized 1 plus 3 equal to 4, which is diabetes mellitus type 1 associated with HLA-3 and 4. Okay, HLA-DR4 associated with type 1 diabetes. And then in addition to this, rheumatoid arthritis and Addison disease. And there is another mnemonic. There are 4 walls in one room. 4 walls in one room. Room represents rheumatoid arthritis. 4 represents HLA-DR4. There are 4 walls in one room. HLA-DR4 associated with rheumatoid arthritis. Okay, that's all for this podcast. Thank you for listening. Best of luck.